So this was a procedure that Dr. Yamanaka and his group developed, several years ago. And it was actually a very elegant way that he identified the procedure to reprogram somatic cells to become pluripotent-like in their phenotype. And so basically what he did is he identified, we have a laundry list of transcriptional regulators that we know are involved and highly expressed in pluripotent stem cells, embryonic stem cells in particular. And so he went in, or his group went in and tested them in groups of 29 at a time and identified several critical transcriptional regulators that can, when overexpressed in a somatic cell type, can convert them to a pluripotent stem cell-like phenotype so that they, in many ways, in most ways, are reminiscent of embryonic stem cells. And he initially performed this in mouse embryonic stem cells, or in mouse somatic cells, to create mouse-induced pluripotent stem cells. And then sure enough, they applied the same transcriptional regulators to human pluripotent stem cells, and then they were able to achieve the same goals. And so there are different cocktails of transcriptional regulators that you can force to express in somatic cell types. And that's sort of the classical cocktail or a combination of OCT4, SOX2, KLF4, and MYC, right? That's sort of the classical ones.
And NANOG?
NANOG sometimes people will throw in there. It's not necessary, but it can work as well. There's a couple, LET1, I think, or LET6, you can also add in there. So there's different little cocktails, and there's also little tweaks people have done over the years to either reduce that or get rid of some of them for different reasons, based on their needs. And so, basically they've shown that, with these same or similar cocktail of transcriptional regulators, you can direct a variety of mammalian cell types to a pluripotent stem cell-like phenotype, as long as you understand and know the culture conditions of the species pluripotent stem cells that you will ultimately want to generate.